myself Sonam Pahuja. Today I will be taking your live session for week 8, that is cell biology, cellular organization, division and cellular processes. So week 8 syllabus covers the cell diversity, properties of specialized cells, budding yeast as a model system, the plant cells, stem cells and nerve cells. So starting from the question number 1, what is the primary function of specialized cells? General metabolism, specific task within an organism, random activities, or none of the above. Anyone wants to answer? Please speak up, don't write in the chat box as I won't be able to see the chat box while presenting my screen. So if anyone knows the answer, please speak up. What is the primary function of specialized cells? Is it general metabolism, specific task within an organism, random activities, or none of the above? Anyone wants to answer this question?
B option, ma'am.
B option, specific task within. Yes,
Yeah, yeah.
yes, right. It's B. So, can you explain the answer?
Like RBC have a function of transport of oxygen, like that, specialized cell.
Yes, you can somewhat relate it to that. And other specialized cells?
Um,
Any other example? But we are going to cover today. Nerve,
um,
nerve cells, right?
yeah, yeah. What?
Nerve cells.
Yeah.
So, the primary function of specialized cells is specific task within an organism, as they are designed to perform particular roles, unlike general metabolism or random activities. These are the cells that have evolved to carry out specific functions within a multicellular organism rather than performing general metabolic processes. Like you have already told that RBCs are specialized for oxygen transport, and we have nerve cells which are specialized for transmitting signals. Muscle cells are also specialized for movements. So all these cells are specialized for some or other task. So these are the specific tasks within an organism which are covered under this. So specialized cells work together in groups to form different tissue types such as muscle tissue or nerve tissue. And then tissues in turn combine to form organs, and these organs work together in systems to carry out the functions of the entire organisms. So these special cells are important because they carry out the specific functions that allow the body to maintain homeostasis or a stable internal environment. Okay, moving on to next. Which organism is commonly used as a model system to study cell diversity? Anyone?
Budding yeast, ma'am.
Budding yeast, right? Although these E. coli, Drosophila, or Arabidopsis are also model organisms to study in particular like bacterial related work or investigation of basic mechanism of molecular genetics for E. coli, and to analyze the effects of genetic mutations or to express and purify particular proteins, to explain also for the gene regulation mechanism including operons and other control systems. And also Arabidopsis in plant systems work very well. So here we want to study the cell diversity. So the most common organism which is commonly used, it is budding yeast. Okay, it is Saccharomyces cerevisiae. Next, what distinguishes a plant cell from an animal cell?
B, ma'am.
Presence of a cell wall. Right. Mitochondria is present in both, nucleus is present in both. So it is a cell wall which plant cells have, but animal cell lacks it. What is the function of a cell wall? Especially in plants, why do they need it?
They provide a rigid structure and protection to the cell, ma'am.
Yes, they provide support and protection to the plant cell. Which organelle is responsible for photosynthesis in plant cells?
Chloroplast, ma'am.
Chloroplast. Correct. What is the defining characteristic of stem cells?
Option A, ma'am.
They can self renew. Right. It means that they can divide and create more stem cells, which also have the capacity to differentiate into various specialized cell types. So, stem cells have the property of self renewal. They have this unique ability to replicate themselves, ensuring a continuous supply of stem cells within the body. They can also differentiate into various specialized cell types such as muscle cells, blood cells, or brain cells or nerve cells, depending on the signals they receive. Unlike other cells, stem cells are also unspecialized, means they don't have a specific function in the body. So, from these options, if we can say they cannot divide, so stem cells have the ability to divide and self renew. So, this is incorrect. They only form one type of cell. This is also incorrect because they have the potential to differentiate into various cell types, and they are non-functional. This is also incorrect as stem cells play a crucial role in development, tissue repair, and also regeneration. So this is the defining characteristic of stem cell, that they can self renew. Moving on to next, which type of stem cell can develop into any cell type in the body? Which type of stem cell can develop into any cell type in the body? Is it pluripotent, unipotent, multipotent, or bipotent? Anyone? Anyone knows this answer? Which type of stem cell can develop into any cell type in the body? Is it pluripotent, unipotent, multipotent, or bipotent? Yes, what? Pluripotent. Yes, it is correct answer. So pluripotent stem cells, they can give rise to all cell types of the body. Okay. What is unipotent? What is unipotent? Multipotent? How many types of stem cells we have? Can anyone explain? We have totipotent stem cell, pluripotent stem cells, multipotent, oligopotent, or unipotent. Totipotent is also known as omnipotent. So these five type of stem cells is on the basis of differentiation potential they have. Okay. So first of all, totipotent or omnipotent, if we talk about, These stem cells are the most powerful that exist. They can differentiate into embryonic as well as extra embryonic tissues. Okay, such as chorion or yolk sac, amnion or allantois. Whatever we have that in humans and other placental animals, these tissues form the placenta. The most important characteristic of a totipotent cell is that it can generate a fully potential or functional living organism. The best known example of this totipotent is a fertilized egg, which is formed when a sperm and egg unite to form a zygote. So it is at or around 4 days post fertilization that these cells begin to specialize into pluripotent cells, which, as described, are flexible cell types but cannot produce an entire organism. Okay, so this is totipotent or omnipotent. Now, if we talk about pluripotent, these are also next most powerful type of stem cell. The importance of this cell type is it can self renew. This one, pluripotent. These can self renew and differentiate into any of the three germ layers. Three germs layer, what we have: ectoderm, endoderm.
Ma'am, I can't see your screen, ma'am.
Is it visible now?
Yeah.
Okay, sorry for the trouble. So these we have these five type of stem cells. Okay, these are based on the differentiation potential. Okay, first of all, I have talked about totipotent or omnipotent, which I had talked that these can differentiate into embryonic or extra embryonic tissues like chorion, yolk sac, amnion, or allantois. And example, best known example of this is a fertilized egg. Okay, now coming to pluripotent, this uh, the importance of this cell type is it can self renew and differentiate into any of the three germ layers. Three germ layers में क्या होगा? Ectoderm, endoderm and mesoderm. These three germ layers further differentiate to form all tissues and organisms and tissues and organs within a human being. So all these three germ layers further differentiate to form all tissues and organs within a human being. So there are several known types of pluripotent stem cells. Also, we know that there is induced pluripotent stem cell term, IPA cells, if you have heard of this term. And also, they are natural pluripotent stem cells. So, if we talk about natural and induced pluripotent, so natural is embryonic stem cell. Embryonic stem cells are unique cells that exist in an early stage embryo. Okay, and if we talk about induced pluripotent stem cell, they were first produced from mouse cells in 2006 and in human cells in 2007, and they are tissue specific cells that can be reprogrammed to become functionally similar to embryonic stem cells. So, because of these powerful abilities to differentiate in a wide diversity of tissues and their non controversial nature, induced pluripotent stem cells are well suited for use in cellular therapy and regenerative medicines. So, this is all about the pluripotent stem cells. Now, if we talk about multipotent, these are a middle range type of stem cell. They can self renew and differentiate into a specific range of cell types. So, example of this is mesenchymal stem cell. MSC, mesenchymal stem cell. These can differentiate into osteoblast. Osteoblast, monocytes, adipocytes, and chondrocytes. Okay, so these are covered under mesenchymal stem cells, which is multipotent stem cells. So these can mesenchymal stem cells can differentiate into osteoblast, which is a type of bone cell, myocytes, which is a type of muscle cell, adipocytes, which is type of fat cells, and chondrocytes, which is a type of cartilage cells. So these cell types are fairly diverse in their characteristics, which is why mesenchymal stem cells are classified as multipotent stem cells. Okay, because they can differentiate into specific range of cell types. तो ये हमारा मेसेनकाइमल में था. Now coming to oligopotent. These are similar to multipotent, but they become further restricted in their capacity to differentiate. Okay, these cells can self renew and differentiate, but they can only do so to a limited extent. They can only do so into closely related cell types. So इसका एग्जाम्पल जो है उसका मेसेनकाइमल स्टेम सेल्स था, इसका हेमेटोपोएटिक स्टेम सेल. दिस इज HSC, hematopoietic stem cell. So these are the cells which are derived from mesoderm that can differentiate into other blood cells. Specifically, जो ये hematopoietic stem cells हैं, these are oligopotent stem cells. These can differentiate into both myeloid and lymphoid cells. ये हमारे ओलिगोपोटेंट में हेमाटोपोइटिक स्टेम सेल्स हैं. एंड हेमाटोपोइटिक सेल्स स्टेम सेल्स जो हैं, these are derived from mesoderm that can differentiate into other blood cells. Specifically, these are oligopotent stem cells which can differentiate into both myeloid and lymphoid cells. Myeloid cells include further basophils, dendritic cells, then inophils, erythrocytes, macrophages, then we have megakaryocytes, we have monocytes, neutrophils, and platelets. These are all myeloid. Now if we talk about lymphoid, these are B cells, T cells, and natural killer cells. सारे माइलॉइड के हो गए एंड दीज ऑल आर lymphoid. Okay. Unipotent. Finally, we have these unipotent stem cells, which are the least potent and most limited type of stem cells. An example of this stem cell is muscle stem cell. Okay. So while as we know that muscle cells can self-renew and differentiate, they can only do so into a single cell type. They are unidirectional in their differentiation capacity. Okay, so the purpose of these stem cell categories is to access the functional capacity of stem cells based on their differentiation potential. Importantly, each category has different stem cell research applications, medical applications, or drug development applications. So, these five different type of stem cells based on their differentiation potential: totipotent or omnipotent. The most powerful among these five, these can develop into a fully functional living organism. An example is fertilized egg. Pluripotent, these can self renew and differentiate into any type of any type of three germ layers: ectoderm, endoderm, or mesoderm. Endoderm or mesoderm. Okay. So these are well suited for use in cellular therapy and regenerative medicines. An example of this is embryonic stem cell. Okay. Now multipotent. These can self renew and differentiate into a specific range. इसमें any of these three germ layers, इसमें specific range of cell types. These will be oligopotent will be closely related cell types. Unipotent will be single cell type. So multipotent, these can differentiate into specific range of cell types. Example, mesenchymal stem cell, and we talked about osteoblast, myocytes, adipocytes, and chondrocytes in these. Now oligopotent stem cells. So these can differentiate into closely related cell types like hematopoietic stem cells, and these are either myeloid or lymphoid. Myeloid covers basophils, dendritic cells, ionophils, erythrocytes, macrophages, megakaryocytes, monocytes, neutrophils, or platelets, while lymphoid covers B cells, T cells, natural killer cells. Now unipotent, these are least potent and most limited type of stem cell, and they can only self renew and differentiate into single cell type. Example, muscle stem cell. Okay, I hope it is clear to all. Is it clear? Should we move ahead? Any doubts till now? So, moving ahead, is the screen visible? So, which technique is commonly used to study live stem cells? Anyone knows this answer? Which technique is commonly used to study live stem cells? Anyone knows this answer? Anyone knows this answer? Which technique is commonly used to study live stem cells? So, answer of this question is live imaging. So, we have different techniques of live imaging of stem cells. Recent advances in live imaging technology have allowed the direct visualization and tracking of a wide variety of tissue resident stem cells in their native environments over time. So, results from these studies have revealed that long standing debates about stem cell regulation and function. So, we have different techniques of live imaging in these. One example is like we have embryonic stem cells, which are typically isolated from inner cell mass of blastocytes using methods like mechanical dissection, laser dissection, or immunosurgery. And while we have adult stem cells, which are derived from tissues like bone marrow and adipose tissue. So these require techniques such as aspiration, harvesting, and specific dissection related techniques. And for stem cell research, particularly, some technologies are frequently used, including stem cell culture, separation, and identification techniques. At present, if you have heard of FACS and MACS, like fluorescence activated cell sorting and magnetic activated cell sorting, so these are common separation methods for stem cells. So moving on to next, which gene editing tool is frequently used to modify stem cells?
Ye-
A. CRISPR-Cas9, right. So all other techniques are just basic techniques or molecular techniques like PCR, Western blot, or ELISA. CRISPR-Cas9 is a genome editing tool which is frequently used to modify stem cells, or in fact in plant genome editing or animal genome editing also, and also humans. So CRISPR-Cas9 is frequently used to modify these stem cells. Can you give some example related to this if you know any common example? Any common example of stem cells which is gene edited through CRISPR-Cas9?
No idea, ma'am.
Okay. Any kind of stem cell, like particularly which type of stem cell can be used to edit in case of humans if we talk about? Out of those five type of stem cells. So most commonly, uh, we used to edit the genomes of human pluripotent stem cells because they have the potential to differentiate into various cell types. So these are the like pluripotent stem cells are most commonly used for the editing purpose. And CRISPR-Cas9 is relatively simple and cost effective compared to other genome editing tools. And it is also used for gene therapy applications. Also, the first use of CRISPR-Cas9 for gene modification in human cells was particularly of hematopoietic stem cells for treating transfusion dependent thalassemia or severe sickle cell disease also. Okay. Moving on to next. Which part of a neuron transmits electrical signals? Dendrites, axon, cell body, or nucleus? Which part of a neuron transmits electrical signals? Is it dendrites, axon, cell body, or nucleus?
I guess dendrites.
Dendrites. For transmission
Yeah.
purpose. It is axon. As we know that axon is a long thin fiber that extends from cell body of a neuron, and it transmits electrical signals known as action potentials. away from the cell body to other neurons, muscles, or glands. So, axon is often covered by a myelin sheath, which acts as an insulator and speeds up the transmission of electrical signals. While dendrites, these are branched and tree like structures, they receive the signal from other neurons. They transmit signals towards the cell body. They does not transmit electrical signals as the axon do. Okay, they transmit signals towards the cell body. So if we talk about electrical signal, it should be complete, like it should be either away from the cell body or towards the cell body. So axon delivers or transmits the electrical signals away from the cell body to other neurons, muscles, or glands, while dendrites, they transmit signal towards the cell body. Okay. And cell body contains the nucleus and other organelles necessary for neurons survival and function. It integrates signal received from the dendrites and transmits signals down the axon. While the nucleus, it is the control center of the cell and containing the cell's genetic material. Next, what is the role of myelin in nerve cells? What is the role of myelin in nerve cells? Is it slows down the signal transmission, enhances signal speed, protects DNA, or helps in protein synthesis?
B, ma'am.
Enhances signal speed.
Yeah.
Yes, it is correct. So myelin primary primary role is to enhance the signal speed because it acts as an insulator. It allows the electrical impulses to jump between the gaps in the myelin sheath. Okay. Myelin is a fatty insulating substance that surrounds the axons of many neurons and it enhances signal speed. It acts like the insulation around the electrical wire, preventing the leakage of the electrical signal. And myelin sheath isn't continuous. There are gaps known as, what are the gaps known as? Nodes of Renvier. Okay. And in myelinated axons, the electrical signal jumps from one node of Renvier to the next, a process known as saltatory conduction. This jumping allows the signal to travel much faster than in unmyelinated axons. You might have heard of that there are myelinated axons and unmyelinated axons. So in myelinated axons, their electric signal jumps from one node of Renvier to the next, and in unmyelinated cells, the signal has to travel continuously along the axon. So myelination is crucial for rapid and efficient communication between nerve cells, and this is essential for various bodily functions including reflexes, movement, and thought processes. And damage to this myelin sheath or demyelination can lead to slowed or disrupted signal transmission, potentially causing neurological disorders. So basically, it increases the speeds by which electrical signals travel along the axons and facilitates neuronal communication between distant regions of the nervous system. Next, what is the primary function of specialized? It has been covered. It is repeated. Repeat. Just the questions are in different format, they are repeated. Which structure in nerve cells helps in fast transmission of electrical signals? It is myelin sheath, okay. What is the role of synaptic vesicles in neurons? Anyone? What is the role of synaptic vesicles in neurons? Anyone? Anyone knows the answer? What are synaptic vesicles? What are synaptic vesicles? So, synaptic vesicles are small membrane bound sacs. These are located in the axon terminals or the end of the neuron, and they are filled with neurotransmitters, which are chemical messengers that neurons use to communicate with each other. So, when an action potential or a nerve impulse reaches the axon terminal, it triggers the release of neurotransmitters from synaptic vesicles into the synaptic cleft, which is the space between two neurons. So, why is this important? The, as we know that released transmitters further they bind to receptors on the receiving neuron, and they can cause a signal to be transmitted, either excitatory or inhibitory. And other functions of these synaptical vesicles is in recycling the membrane after exocytosis and refilling with neuron transmitters. So the role of synaptic vesicles in neurons is which of these? Can anyone tell now? They store the neurotransmitters that are released at the synapse. So they play central role in synaptic transmission. They are also regarded as key organelles which are involved in synaptic functions like uptake, storage, and stimulus dependent release of neurotransmitter. Moving on to next. Which type of glial cell is responsible for immune defense in the central nervous system? Which type of glial cell is responsible for immune defense in the central nervous system?
He occurs.
Astrocytes, microglia, Schwann cells, or ependymal cells? Anyone knows the type of these glial cells? No one? So the answer is microglial cells. So the glial cell which is responsible for immune defense in the central nervous system, it is microglia. These are resident immune cells of the CNS which acts as the brain's first line of defense against pathogens and other threats. While we talk, if we talk about astrocytes, these are primarily involved in the structural support. Structural support. Nutrient transport. And maintaining the blood brain barrier. Astrocytes. Astrocytes are involved in structural support, nutrient transport, and maintaining the blood brain barrier. Now, if we talk about Schwann cells, these are found in the peripheral nervous system and not in the central nervous system. This is present in CNS. These cells form the Schwann cells. These form the myelin sheath.
Ma'am, I can't see your screen, ma'am.
Sorry again. Okay. Myelin sheath. These These Schwann cells are present in peripheral nervous system, and they are responsible for formation of myelin sheath around the axons, which is not present in the CNS. Okay. Now, if we talk about ependymal cells, these line up the ventricles of the brain. These line up the ventricles of the brain, and also these produce CSF. What is CSF? Cerebrospinal fluid. Okay. And it asked the question asked about the glial cell which are responsible for immune defense in the CNS. So this is microglia. This is the first line of defense in the brain which protects against pathogen defense against pathogen and other threats. Okay. And this is present in CNS. I hope it is clear now. Which plant organelle contains chlorophyll? It has been covered already. Huh. Which molecule is the primary neurotransmitter at neuromuscular junctions?
C, ma'am.
Can you repeat?
C.
C. Acetylcholine. Right. What is the function of other molecules? Neuromuscular junctions are the sites where motor neurons communicate with the skeletal muscle fibers and they trigger muscle contraction. So here acetylcholine is released by the motor neuron into synaptic cleft. Synaptic cleft क्या होता है? The space between the neuron and the muscle fiber. And then acetylcholine binds to the receptors on the muscle fiber membrane, and it leads to the opening of ion channels which initiate the muscle contraction. While talking about other neurotransmitters like these, serotonin is involved in the mood regulation, sleep, and digestion. Dopamine is involved in reward, motivation, and movement. Acetylcholine is what we talked about, that it is present in neuromuscular junctions and involved in the muscle contraction. And glutamate, it is the primary excitatory neurotransmitter. It is excitatory neurotransmitter in the brain. Next, what is the function of hemidesmosomes in cells? What is the function of hemidesmosome in cells? the answer? Anyone knows the answer? What is the function of hemidesmosomes in cells? No? So, the primary function of hemidesmosomes is to attach cells to the extracellular matrix, specifically connecting the basal surface of epithelial cells to the underlying basement membrane and extracellular matrix. So, these are specialized cell junctions. They are distinct from other cell junctions like desmosomes, which connect the neighboring cells. These anchor cells to the extracellular matrix. This is achieved by linking the intracellular keratin intermediate filaments to the extracellular matrix proteins. Hemidesmosomes are particularly important in basal epithelial cells. Hemidesmosomes are particularly important in basal epithelial cells, where they contribute to the stable adhesion of these cells to the underlying basement membrane. So, these are particularly important in basal epithelial cells. They contribute to the stable adhesion of these cells to the underlying basement membrane. And also, by anchoring cells to the extracellular matrix, these hemidesmosomes play a crucial role in maintaining the structural integrity and strength of the epithelial cells. And these are found in tissues like skin, cornea, and parts of gastrointestinal and respiratory tract and the amnion. And these are not just adhesion structure, they are also involved in signaling pathways such as keratinocyte migration or carcinoma cell intrusion. Okay, so these are specialized junctional complexes that contribute to the attachment of epithelial cells to the underlying basement membrane in stratified and other complex epithelia such as skin, cornea, or parts of gastrointestinal, respiratory tract, or amnion. Okay, these are multi-protein complexes that facilitate the stable adhesion of basal epithelial cells to the underlying basement membrane. And the mechanical stability of hemidesmosomes relies on the multiple interaction of few protein components that form a membrane embedded tightly ordered complex. So these connect cells to the extracellular matrix. Which of the following is not a function of stem cell? Anyone?
Option C, ma'am.
Option C. Correct. So we already learned about that stem cells are involved in self renewal and differentiation, and also Renewal will happen if any damage happens to any cell. So it also replaces the damaged cells. While producing antibodies is not a function of the stem cell. Which of the following ions is primarily responsible for depolarization in neurons?
B, ma'am.
Yes, correct. You know polarization and depolarization of neurons? So depolarization, it refers to the process where the inside of a neuron's membrane it becomes less negative or more positive compared to its resting state. Here the role of sodium is when a rapid influx of sodium ions into the neuron happens through voltage gated sodium channels, it causes this membrane potential to become more positive, which initiates the depolarization. Okay, while potassium plays a crucial role in repolarization, that is, returning the membrane potential to its resting state. And calcium and magnesium, these two, these are responsible or have role in neuronal function. They're not primarily responsible for depolarization. So in neurons, the rapid rise in potential or depolarization is an all or nothing event that is initiated by the opening of sodium ion channels within the plasma membrane. So the subsequent return to resting potential or repolarization is mediated by the opening of potassium ion channels. Okay, so voltage gated sodium channels are responsible, which carries the influx of sodium ions and it gives rise to depolarization. And while increased potassium accumulation, it results in as we know that sodium influx responsible for membrane depolarization and potassium crucial in repolarization. It returns the membrane potential to resting state. So these are the questions which have been covered. Now talking about some concepts.
Thank you, ma'am.
Now I will cover the some concepts relating to today's syllabus only, like stem cells. What was the syllabus? Cell diversity and properties of specialized cells, like budding yeast as a model system. So first of all, we will talk about budding yeast, which is a model system. So why it is a powerful model system for studying eukaryotic cell biology? What are the advantages budding yeast has over other organisms to be a model system? First of all, ease of manipulation. So as we know that budding yeast is easy to grow and manipulate in the laboratory, both in haploid and diploid forms. So it is easy to manipulate. Second, it has rapid growth. Its short generation time allows the rapid experimentation and observation of cellular processes. Third, well characterized genome. So budding yeast, it is the first eukaryote to have its entire genome sequenced, producing a rich resource for genetic and molecular studies. So many fundamental cellular processes including cell division, DNA replication and protein synthesis all are highly conserved between yeast and other eukaryotes including humans. Budding yeast has a robust homologous recombination system facilitating genetic manipulation and creation of mutants. So it is kind of versatile tool or genetic or biochemical tool which has been developed for studying yeast, enabling researchers to investigate various cellular processes in detail. Also, it is non pathogenic. So, it is non pathogenic, making it safe to work within the laboratory. It is a model for aging. So, it has emerged as a model organism for studying aging, with its replicative aging thought to be comparable to aging phenomena observed in higher eukaryotes. So, it is used for as a model for aging. It is a model for aneuploidy. Okay, it is a robust and versatile model system for studying the effects of genetic alterations, particularly aneuploidy, due to its small genome and tolerance of aneuploidy. And now covering about areas of research. So first of all, genetic inheritance. Next is cell cycle, metabolism, protein trafficking, aging, human disease. So these are the areas of research. Talking about genetic inheritance, Studies in yeast have contributed significantly to our understanding of gene expression, DNA repair, and other aspects of genetic inheritance. Cell cycle. It has been instrumental that budding yeast is used in understanding the cell cycle, including DNA replication, mitosis, and meiosis. In metabolism, it is a valuable model for studying metabolic pathways, particularly those involved in fermentation and energy production. Protein trafficking. It has been widely used to study the mechanism of protein trafficking, including the synthesis, transport, and localization of proteins. Aging. It is a model organism for studying the mechanism of aging and effects of aging related factors. And in human disease, it leads to insights into the molecular basis of human disease, including cancer, neurodegenerative disorders, and birth defects. Okay, and yeast is used in several molecular to study several molecular pathways and also in molecular techniques like yeast to hybrid assays for studying protein protein interaction, split ubiquitin assays, yeast complementation assays, and several others. Yeast 1 hybrid assay also, like studying DNA protein interaction. Okay, now talking about the plant cell. What is the difference between plants and cell and animal cell?
It has cell wall um, and chloroplast.
Yes. Any other difference?
Also, the presence of vacuoles.
Yes.
Um,
Yes. So
It
plants.
produces their own food from photosynthesis by chloroplast.
Yes, true. What about centrioles? Centrioles, lysosomes. Do they have? No idea.
Yeah, I think yes, ma'am.
So plant cells have a cell wall, which is a rigid outer layer made of cellulose that provides the support and protection, which animal cells don't have. Okay, animal cells have cell membrane. यहाँ पे हमारी सेल वॉल है, यहाँ सेल मेम्ब्रेन है. Cell wall, rigid outer layer made of cellulose. Cell membrane, it is a flexible outer boundary that regulates what enters and exits the cell. Okay, now another difference is chloroplast. these are the organelles where photosynthesis takes place, allowing plants to produce their own food. Okay, while these are absent in animals. Another is a large central vacuole. So, in plant cells, we have a large water filled sac that helps maintain cell shape, stores water and nutrients. We have other plastids as well. Organelles involved in various functions including storage and pigment production. These have plasmodesmata, which are the channels that allow communication and transport between plant cells. And then centrioles and lysosomes. Plant cells do not have centrioles, which are involved in cell division, and lysosomes, which are involved in waste removal. Okay, centrioles in cell division and lysosomes, they contain enzymes which are responsible for breaking down the waste and cellular debris. Also, animal cells do not have cell wall, chloroplast, or large central vacuole. They have small vacuoles but not as prominent as in plant cells. Okay, then talking about stem cells. What are stem cells? Most of it is covered where I have discussed about the types of stem cells based on the differentiation potential. So, stem cells, these are undifferentiated cells with a remarkable ability to develop into various specialized cell types, offering the potential for regenerative medicine and disease treatment. So these are characterized by their ability to remain in indifferentiated state, meaning they haven't yet become specialized into a particular cell type, and they possess the capacity to differentiate into various cell types depending on the signals they receive, like differentiation potential. So we have different types of stem cells, like embryonic stem cells. They are derived from early embryos. These cells are pluripotent. They can differentiate into any cell type. Then we have induced pluripotent stem cells. These are adult stem cells which are reprogrammed into a pluripotent state and offers a potential source of stem cells without ethical concern, as they are induced pluripotent stem cells. Then we have adult stem cells. These are found in various tissues. These cells are generally multipotent or unipotent, meaning they can differentiate into a limited range of cell types. So earlier we talked about differentiation potential. Earlier, these are the types of stem cells based on their differentiation potential. Based on their differentiation potential of stem cells, we have five types of stem cells: totipotent or omnipotent, pluripotent, multipotent, oligopotent, or unipotent. We have discussed about these already. So now we have types of stem cells like embryonic stem cell, then we have induced pluripotent stem cell, and we have adult stem cell. So these are different kind of these only. Like embryonic stem cells are pluripotent, we have already talked about, as they differentiate into germ layers, ectoderm, endoderm, or mesoderm, and can form full organism. They can differentiate into any cell type. Now induced pluripotent stem cell, these are reprogrammed to a pluripotent state and they offer potential source of stem cells without ethical concerns. Then we have adult stem cells which are found in various tissues. These cells are generally multipotent. Adult stem cells, these are generally multipotent or unipotent, meaning either these mesenchymal or muscle stem cell. Okay, they can differentiate into a limited range of cell types. Role of like many people do stem cell research, and what are their applications? Basically, in accelerating research, in improving stem cell manufacturing, then we have enhancing cell characterization, optimizing stem cell therapies, and then drug discovery. So, if I talk about stem cells, these can be gene edited using different approaches of genome editing tools, and we have different live imaging techniques which are used to study the cell biology. So we have cell labeling strategies and detectors for stem cell imaging. Like if we have for direct labeling cells are incubated with different imaging probes that enter the cell via transporter uptake. We have different imaging probes. This is for direct labeling. Okay. So for direct labeling, cells are incubated with different imaging probes that enter the cell via transporter uptake and endocytosis or passive diffusion. So for different mechanism we have different imaging probes. Okay. If I talk about reporter gene, we have reporter gene imaging. So here cells are transfected or transduced with reporter gene construct. And transcription of the reporter gene is under the control of a promoter, which is followed by the translation of its mRNA, leading to the accumulation of different reporter proteins such as receptors, enzymes, or transporter proteins. An introduction of this reporter gene probe results in a signal generation. Now, these labeled cells are detected via various imaging systems. We have different imaging systems like PET scan, if you have heard about, MRI, or CT scan, or ultrasound. So these are various imaging systems where labeled cells are detected. Now we have different imaging techniques for cell therapies. Like we have bioluminescence imaging, we have FMT, which is fluorescence tomography, we have ultrasound, then we have SPECT, then we have PET, then we have MRI and CT. So if I talk about one by one everything, like bioluminescence imaging. So it is basically for preclinical only, and acquisition time is in seconds. Then it is, the strategy behind the labeling is reporter gene, and it has advantages like it is cheaper, simple, high throughput, and disadvantages are it can cover only small animals, low resolution, and only 2D images are covered here. low resolution, and then we have only 2D images in this bioluminescence imaging. Then we have FMT, fluorescence tomography. This is also reporter gene based and also fluorescent dye based. It is cheaper, simpler. It also has low resolution and cells need to be close to the surface. It is also low resolution and cells need to be close to the surface. Now ultrasound. It is reporter gene as well as antibody with microbubble. It is cheaper, relatively simple. It has limited 3D capabilities. And it has low signal to noise ratio. Next, SPECT, single photon emission computed tomography. Okay, single photon emission computed tomography. It is reporter gene and incubation with radio tracer. It covers 3D imaging. And here radioactive tracer is required. Also anatomic reference is also required. Then we have PET, positron emission tomography. It is reporter gene and it is also incubation with radio tracer. Involves 3D imaging but with high resolution. 3D imaging with high resolution. Here also radioactive tracer and anatomic reference required. Now MRI, magnetic resonance tomography. It is 3D imaging only, good with soft tissue, no radiation, high resolution. So it involves internalization of surface labeling with nanoparticles or specific ions. It is very expensive and complicated. Although it involves high resolution 3D imaging, no radiation are involved, and it is good with the soft tissue as well. Now CT scan, which is computed tomography. It is internalization of surface labeling with nanoparticles. It is also involved for 3D imaging, relatively cheaper and high resolution. It uses ionizing radiation. So these are the different techniques. These are the imaging techniques for cell therapies. So, if I talk about barriers to clinical translation, we have a variety of stem cell types. In example, we have adult somatic cells, induced pluripotent stem cells, or embryonic stem cells. All are available for transplantation, each with their advantages and disadvantages. But adult somatic stem cells, which are most commonly transplanted directly into the recipient, whereas embryonic and induced pluripotent stem cells, they undergo in vitro differentiation prior to transplantation. Regardless of the stem cell type, their successful application in the regenerative therapy faces similar clinical hurdles, including limited engraftment, survival, and proliferation, poor differentiation, maturation, and integration, immunogenicity for allogenic transplantation. Then we have potential tumorigenicity with pluripotent stem cell derivatives. So, cell imaging plays a pivotal role in overcoming these hurdles, which will help guide the translation of this promising therapy. Okay. What else we can cover? So, if I talk about step by step approach to successful routine application of stem cell therapy, first of all, it is stem cell isolation and purification. First of all, stem cells must be initially isolated and purified from a tissue source. Because of poor in vivo differentiation, cells are often differentiated in vitro prior to implantation. So, these techniques, however, are imperfect and can result in a mixture of undifferentiated and differentiated cells. So, cell sorting is then required to select for the cell population that can be safely transplanted with maximal therapeutic benefit. First is stem cell isolation and purification. So, I have already talked about this. Then we have cell labeling and transportation. So, prior to transplantation, cells are labeled to track cell fate. Selected cells are labeled in vitro using direct labeling or reporter gene techniques and then implanted at the bedside via intravenous delivery or in the operating room or angiography suite via intralesional or intraarterial delivery. Then we have serial imaging. So we have serial imaging for safety and efficacy monitoring. So after delivery, labeled cells are tracked using PET or MRI, which provides high sensitivity for for cell detection as well as excellent spatial localization. So cells can be tracked serially to determine whether they engraft, survive and proliferate in the target organ or not. So cells can also be monitored for any potential unwanted effects, including tumor formation, migration to other sites. In addition, PET and MRI can provide serial data on the functional and structural recovery of damaged tissues. So this is step by step approach of stem cell, and here imaging is required. So if I talk about this gene editing techniques, we have different gene editing techniques, but now most commonly CRISPR-Cas is used widely. We have different gene editing tools like ZFN, zinc finger nucleases, talents, transcriptional activator like effector nucleases. Then we have CRISPR-Cas nucleases. We have different base editors like cytosine base editor, adenine base editor. We have different prime editors, prime editor 1, 2, 3, 4, 5. Then we have epigenetic editors, transcriptional repressor or transcriptional activator. Okay, so these are different gene editing tools required for stem cell editing or therapy. Then we have nerve cells. Nerve cells are also known as neurons. They are fundamental units of nervous system and they are responsible for transmitting electrical and chemical signals which enables communication between the brain, spinal cord and body. So we have dendrite, soma, axon, node of Renvier, axon terminal, Schwann cell, myelin sheath and nucleus inside the neuron. Yes, in exam MCQ type questions will be asked, but I don't have detail about that negative marking. I will let you know in the next extra class. Okay. So talking about types of nerves, we have sensory nerves, motor nerves, mixed nerves. We have different nerve disorders as well. So functions of these nerve cells are receiving and processing sensory information. These receive signal which can be electrical or chemical from other neurons or sensory receptors. Then they process this information determining whether to transmit the signal further. Then transmitting signals. These transmit signal also called as nerve impulses to other neurons, muscles or glands. So this transmission occurs through a process known as synaptic transmission, where neurotransmitters or chemical messengers are released across a tiny space called a synapse. Further, we have axon, which is a long cylinder projection of the neuron which transmits the signal to the synapse. Then we have dendrites, branched extensions of the neuron which receive signals from other neurons. The neurons can receive signals from multiple sources and these can integrate this information to produce a specific response. This integration is crucial for complex behaviors and decision making. And then we have cell body, which is known as soma. We have cell body, axon, dendrites, synapse, and neurotransmitters. So cell body, it contains the nucleus and other essential organelles. Dendrites are branch-like structures that receive signals from other neurons. Axon is a long slender projection that transmits signals away from the cell body. Synapse is the junction where two neurons communicate, and neurotransmitters are the chemical messengers that transmit signals across the synapse. So they can use the electrical as well as chemical signals to send information between different areas of brain as well as between the brain, the spinal cord and the entire body. Okay. Finally, I will cover about the depolarization and repolarization in neuron. So in a neuron, depolarization is the process where the inside of the cell becomes less negative, that is more positive, due to an influx of positively charged ions. While repolarization is the process where the cell returns to its resting negative state as positively charged ions flow out. So depolarization may, it is influx of positively charged ions depolarization में, and repolarization it will be efflux of positively charged ions. So positively charged ions flow out and cell returns to its negative resting negative state. So we have four stages. First is resting state. Then we have depolarization. Then we have repolarization. And finally, we have hyperpolarization. Okay, so in the resting state, Neurons have a resting membrane potential which is typically negative on the inside compared to the outside. So it is negative on inside compared to the outside. The negative charge is maintained by a concentration gradient of ions, specifically sodium and potassium, and the sodium potassium pump. Then depolarization. When a neuron receives a signal, example from another neuron or sensory receptor, it can trigger the opening of voltage gated sodium channels. So here sodium influx होगा. So sodium ions rush into the cell, making the inside of the cell less negative and more positive. Voltage gated sodium channels. If the depolarization reaches a certain threshold, an action potential or a nerve impulse is triggered. And in then repolarization, sodium channels are inactivated. So as the action potential reaches its peak, the sodium channels close and the potassium channels open. So here potassium efflux happens. Potassium ions flows out of the cell, restoring the negative charge inside. Again more negative inside. So the cell returns to its resting membrane potential, ready to fire another action potential. Now hyperpolarization, potassium channels may remain open slightly longer than necessary, causing the cell to become even more negative than its resting potential. So यहाँ पर potassium channels open ही है for a long time. More negative. Then resting potential. This brief period of hyperpolarization is followed by the sodium potassium pump. Again here sodium potassium pump will be involved, which re-establishes the resting state. Okay, so this is the action potential. Depolarization, repolarization, hyperpolarization, and resting state. So the whole cycle or the nerve impulse action potential is generated. I hope it is clear. So thank you everyone for joining.